Now that's true for us, but it's not necessarily the case for organisms that rely on being able to regenerate, truly regenerate, in order to survive. And we're going to have a little look at some of these, because I'm a biologist and I love animals. And we're going to see how simple animals, such as the planaria that you see on the left, on the top, the hydra, which is the green thing that looks like a sprig, and the famous starfish, are capable of regenerating. They regenerate in the most miraculous fashion. You can cut these things in half, and literally both halves will make a new organism, and I'll show you a bit about that. The complex animals you see below also are very good at regenerating, but they do it with a little less drama, in that they can regenerate a limb or a tail, but they can't regenerate an entire animal from just a single chunk. So let's look at planaria. Some of you in the audience have actually done experiments with this as preparation for this talk, and so this will be old hat to you. But a planaria that's cut in half can actually regenerate the part that's missing. That means that the whole head of the planaria can regenerate and the whole tail can regenerate. And this is because planaria are actually full of stem cells. They have little cells called neoblasts in planaria speak, which are capable of regenerating large portions or whole planaria. And so that's the reason why the limit of regeneration in a planaria is so extraordinarily dramatic. Cutting a planaria into many little pieces gives you a planaria for each piece. And that's because each piece contains some of those stem cells. So some of the students did an experiment with planaria, helped by Alejandro Alvarez, who's an expert on this. And the question they had was, in a planaria, shown here with the head on the left, stained up blue, and the tail on the right, if one cuts the planaria in two at either position 1, 2, or 3, leaving an almost whole planaria minus a head, a half a planaria, or just a tail, what happens at the regeneration point? Basically what happens is you get a new head in each case. And you can see this by looking for the eye spots. Those cute little eye spots are actually photosensitive. Now the question was, is a planaria's capability for making a head different if it's here or if you have to start here? And so the ability to do this was tested by looking at the time it took to put that head back into place. And as you can see, the winner of this game was the largest piece, which had a very short time before the head was formed, whereas the loser was the tail. It took a bit longer, but even so, we got a tail growing a head. And here are just some beautiful shots of the students' work in which you see the stem cells, those neoblasts, those sparkly cells, shown up on the right as a stain for the stem cells and on the left in the context of the whole animal. Now it just so happens the one on the left had its tail cut off and the one on the right had its head cut off, and that's why those cells seem to be so active at this point. Okay, so now that's one kind of very dramatic regeneration, but it's hard to relate to a planaria. I mean, it's got cute little eyes, but for the rest of it, it doesn't have much else that looks like us. However, I'm going to show you an organism, if I can put this glove on straight, that looks a lot more like us than that planaria, although you may not think so. For a biologist, this guy is one short step away from mankind. And I'm going to bring him out here and see if I can get him on the close TV. There he is. This is a salamander, or a newt. I'm going to put him here out so that you can see him, I hope, on the screen. He's really quiet at the moment, but these guys can scamper like crazy. And they're very, very small and very easy to lose, but they have, as I hope you can see, a head. He's moving it now. They have 2 forelimbs, 2 hind limbs. They even have fingers. I think these guys have 5 or 4, and a tail. And inside they have a beating heart, lungs, a liver, a pancreas, intestines, a skin, and a very small brain. And they're very, very capable of running and outrunning their prey, but if by any chance part of their tail or their limb gets cut off, they can regenerate the entire thing. So you see, I'm just going to see if I can make him move a little bit so that you can see those limbs. You see how detailed they are? Now Doug was entranced at the rehearsal and asked me to get this little guy, now this didn't happen in the rehearsal, to get this little guy and show you what his belly looks like, because his belly is beautiful and red. Isn't that pretty? Now I'm going to do another trick, which is, he's so cute, I'm going to kiss him and turn him into a prince. That was for you, Doug. Okay, Guy, that's enough for you. Back in the story. Now, he's lucky. He didn't get his leg cut off. He just got a kiss. But what I'm going to show you is what happens if you do cut off the leg of a newt. And for that, we have a beautiful video. So let's start the video. Now, we're going to have a look at the way in which an amputated limb grows over the course of about 90 days in a salamander. That's a time-lapse movie, watching that thing grow. And now we're going to see what's really going on. Here's the salamander, and it's got a completely new limb. It looks perfect. It has inside bone, it has nerves and muscle, and it can even wiggle. In fact, it's perfect. Now we're going to cut it off. But here's the good news. It grows back. So don't be too scared. First, wound healing. Did you see that wound heal? Now we're watching what the newt can do that we cannot do. Cells are streaming out of the surrounding tissues into the area of the wound and forming what we call a blastema, which is a group of undifferentiated cells that are in fact really just like stem cells. And they're multicolored because they come from skin, from muscle, and even from cartilage. And these have a miraculous memory of what they used to be and are able to form a perfectly functional limb. And that happens all within anywhere from 30 to 90 days, depending on the size. Those little guys would do it faster. So we'd like to know more about how that works. One thing that I should say is that unlike the planaria, the limb that's cut off cannot make a new newt. And you might want to know why that is. Because in theory, you could ask, well, maybe you can just clone the nose, as Woody Allen would say. But we can't, and here's why. Let's have a closer look at what's going on when that regeneration blastema, or group of cells at the tip, starts to really do its trick. So there's the amputation plane of a really early stage in which all those cells have moved up. And inside that blastema are cells that are differentiating, that come from a number of different sources within the stump, an epithelium that closes over, dedifferentiated cells that are probably stem cells, and extracellular matrix that hold the whole thing together. Those are the players. Now what we know from some beautiful experiments that were done by Jeremy Brox and Eli Tanaka is that you can label cells from the unamputated limb that are in fact muscle stem cells, and I'm going to tell you a bit more about those in a minute. And those can actually go in, can be re-injected into this amputated stump, and then they will actually begin to form part of the regenerating limb. And if we label those, we can see what they become. And what you're seeing here is that muscle stem cells became cartilage, and in another picture, you see here that muscle stem cells became skin. So this tells you that there is some potency within the regenerating limb that allows cells to make decisions about being things that they normally weren't going to do if this traumatic event hadn't happened. And this kind of plasticity is something that is extremely interesting to biologists, because it's the key to understanding how it is that stem cells in the adult are capable of recapitulating some of that marvelous potency that we found in the embryo. So I'm going to leave you then, before questions, with this question of why we can't regenerate missing body parts. Is it because we're missing certain kinds of signals? Is it because there aren't enough stem cells? What are the reasons? And what we'll hopefully do after questions is then to explore some of the mechanisms that we have discovered in this fast-moving field to solve some of these mysteries.